Okay, so couple quick things. We're going to start now material on chapter 7, which is about, so last lecture we talked a lot about lipids. Well, lipids are going to be a main thing that we're talking with chapter 7 material then as well, because we're going to be talking about membranes. And one of the component of membranes is lipids, or most specifically phospholipids then. So we're going to discuss membrane structure, membrane permeability, fluidity, mosaicism, important functions of the membrane, and then finally transport across the membrane. Again, when we're talking about membranes, though, membranes are the boundary of a cell. Membranes set out the cellular boundary. Things that are outside the membrane are also outside the cell. Things that, and then the cell is the membrane and everything kind of inside of it. That membrane is a protective barrier for the cell. It keeps harmful stuff out. It keeps stuff that might be damaging to the cell out. It is a protective barrier, and you can think of it almost as also a gatekeeper. It controls what can get into the cell, what cannot get into the cell. And not only that, it controls what can get in, but also out of the cell then as well. So it is a gatekeeper, and it is a restrictive thing. It has what is called selective permeability. And permeability is just referring to what can go in and out, but it is selective in what could get in and out of the cell then as well. So these membranes are going to have what's called selective permeability. We're going to talk about why that is the case here. And we're going to talk a little bit about what's the structure of the membrane, and more specifically, what biological macromolecules are found in the membrane as we go through this then as well. Okay. First, one of the major components of membranes, and we talked about this last lecture, is phospholipids. Phospholipids are going to be the main component of membranes. And again, that phospholipid, if we look at its structure, we have the two fatty acid chains, which are largely hydrocarbon chains, carbon-hydrogen chains, and I've got those highlighted in green up here. By the way, am I showing saturated fatty acids or unsaturated fatty acids in this particular image? How many? Let's go show of hands, actually. You guys have to pick one. There's only two options. Should you guys put your hand up if you think that these are saturated fatty acids? Hands up if you think they are. How many think they're unsaturated? These are saturated fatty acids. Saturated fatty acids have no double bonded carbons in them. Are there any double bonded carbons in the green parts here? Nope. All the carbons have single covalent bonds between them. And so that is going to be a saturated fatty acid. An unsaturated fatty acid is going to be any hydrocarbon chain that has then double bonded carbons, two carbons that have a double covalent bond between them. All right, so the fatty acids are highlighted in green. They're largely hydrocarbons. They are then linked onto a glycerol molecule for a phospholipid. I have that is a 3-carbon sugar alcohol, and that is highlighted in the orange. And then there's a polar head group, which is usually got a phosphate group and then a little chemical group attached to that then as well. But I'm going to call that polar head group, but phosphate's always part of that polar head group. This is specifically a polar head group called phosphocholine, phosphate choline. But there are some other things besides choline that can sometimes be at that one position. There's always the phosphate. Sometimes it's usually choline chemical group. Sometimes it's some other stuff. Phospholipids are going to be the main component. And again, if this is one of my individual phospholipids here, this is supposed to be maybe the polar head group and then the two fatty acid tails in yellow. This is then, again, just a blown up of that polar head group, glycerol, two fatty acid tails. And we're seeing a phospholipid from one layer and then from the other layer of a phospholipid bilayer. Again, membranes are mostly composed out of a phospholipid bilayer. With that phospholipid bilayer, the polar head groups are always towards the outside of one layer and towards the outside of the other layer. So again, I have one layer here. I have a second layer here. It's a bilayer. So I got one layer of phospholipids and a second. The polar head groups are always pointed towards the outside of one layer and the outside of the other layer. The fatty acid tails are always pointed inwards towards one another between the two layers. This is largely because, again, when they do this, phospholipids will automatically self-assemble into a bilayer when you have them in an aqueous solution, in a water-based solution. The polar head groups are hydrophilic. Why are they hydrophilic? Because they're charged. They've got that phosphate group with a negative charge. So that charged polar head group likes to interact with the polar water. Remember, water also has partial charges. Partial positive charges on the hydrogens, partial negative charge on the oxygen. So the charged phosphates like to interact with partially charged water. Questions about that? But the fatty acid tails are hydrocarbons. Carbon and hydrogen are held together by what type of covalent bond? A non-polar covalent bond. So they are non-polar or non-charged. So the fatty acid tails are largely uncharged, or what is called hydrophobic. Again, that is because of the fatty acids being uncharged. But the fatty acids don't like to interact by water, but by being in the middle, they're not exposed to water and they don't like to mix with it. The fatty acids do like to interact with other uncharged fatty acids. So the fatty acids from one layer are interacting with the fatty acids of the other layer. But this gives you a fairly substantial region of the membranes, all that part in the middle that is hydrophobic. That thickest part of the membrane, the middle of those two layers, is going to be a pretty substantial hydrophobic layer, uncharged layer. Questions about that? The phospholipids always, if you mix thousands of these into water, they always self-assemble in the structure because of their way they interact with water. And this ends up being the most stable arrangement to it then as well. All right, so membranes are mostly phospholipids, but I said mostly, which means there's some other stuff. And then this brings up what is called the fluid mosaic model of membranes. The fluid mosaic model describes membrane structure and membrane function. So what it is, it's describing two things, but it's describing membranes are fluid. So the first part, the fluid part, is that they are fluid or that they are movable. They are malleable. And that they are dynamic. What's dynamic mean? Something's dynamic versus static. Yeah, dynamic means it's changing. Membranes are not static structures. They're not things that are stuck in place. They're malleable, they're flexible, they move over time. Okay. And that's really important because if membranes weren't malleable, every time you grab something with your hand, you'd be killing the cells in your body. And membranes were not malleable, when I say you went to pick something up like a pin, well, you're squeezing your cells. You're putting physical stress on them. If the membranes weren't malleable, every time you pressed on them, you could break and crack them open, and that would kill the cell. Okay, so membranes got to be malleable. That's part of their protective feature. The other part, though, of the fluid mosaic model is that they are then mosaics. Membranes are mosaics, and a mosaic just means it's built up of multiple different kinds of parts. Multiple different parts are part of the membrane structure. It's not just phospholipids. It's other things as well, is what I'm really trying to get here. And you can see in this particular diagram, I have, for instance, phospholipids. Like there's one of my individual phospholipids that I just circled, but there's a lot of phospholipids in a membrane, but there's all this other stuff, like these proteins that are part of it. Carbohydrates are part of it. Glycolipids, these are sugar fats, sugar lipid molecules. So there's a lot of different parts to these membranes then as well. And I'm going to talk about this fluid mosaic model in more detail here. Okay. All right, just when we're looking at mosaicism, let's talk about actually mosaicism first. The plasma membrane, we can do things with the plasma membrane. For instance, we can freeze fracture it and look at it via electron microscopes. So one of the things that's been done before is they freeze a cell, cryo-freeze it, things like liquid nitrogen, say, for instance. But then using a molecular knife is what is sometimes referred to as, they've been able to take the two layers of the membranes apart. So they can actually look and take this layer apart away from the opposite layer. So they break that phospholipid bilayer apart and they can look at it via electron microscopy. And what they find is that they look at it, they often find these bumps. In particular, they find a lot of time on the cytoplasmic side. They find all these bumps. Well, the phospholipid bilayer would just like basically look nice and thin and straight. It would be pretty uniform. What are all those bumps? Well, they're proteins that are embedded inside the membrane. And you can see some of those proteins embedded into there. And it basically just looks like this. As they take those two layers apart, sometimes the proteins stay in one layer, sometimes the proteins go in the other layer, sometimes you're left with holes where those proteins once were from these frozen membranes. But they can see that, they can look at that, and they can split that bilayer. And they can see there are multiple parts in there. With the proteins, proteins typically come in two different varieties. There are what are called integral membrane proteins. Integral membrane proteins are going to be ones that are integrated into the membrane. In essence, they're embedded inside of it and they pass through the membrane. So in integral membrane proteins, this is my membrane, my phospholipid bilayer. They pass through the membrane and they're inside of it, at least for part of their structure. There are also, though, peripheral membrane proteins, and peripheral membrane proteins are just more loosely associated with the surface of the membrane then. You get different kinds of proteins based upon their structure. Structure always correlates to function. So for instance, with integral membrane proteins, the part of the protein that passes through the membrane is made up of a hydrophobic amino acids with a hydrophobic side group. Things like valine, leucine, alanine. These are amino acids that have basically side groups that are completely just made of carbon and hydrogen that are uncharged, hydrophobic. So those hydrophobic amino acids are the portion that pass through the membrane so that you have hydrophobic amino acids interacting with the hydrophobic regions. Again, likes to interact with like. Uncharged things, uncharged amino acids, like to interact with the uncharged fatty acids. So that's where you get an integral membrane protein. Peripheral membrane proteins kind of tend to lack that nice hydrophobic center, in essence, or something. All right, so this might be, again, an example of a protein passing through a membrane. This is my protein, kind of the structure. This is the N terminus of the protein, the C terminus of the protein. What's the N terminus and C terminus describe again, though? The amino and the carboxyl group. Remember, proteins at the beginning have an N terminus for the amino end or nitrogen end. An amino group has nitrogen in it. They also then have at the end a carboxyl terminus or C terminus, because that's where the carboxyl group is coming. So the N terminus is the beginning of the protein. The C terminus is the end of the protein. But proteins and polypeptide chains always have an orientation. The amino group is going to be coming off the front end. The carboxyl group of a growing protein chain is always coming off of the end. Does that make sense, everybody? What type of protein is being shown here? Integral membrane protein or peripheral membrane protein? Integral, because it's going through the membrane in my diagram here. What type of secondary structure is being shown in my protein here? Yeah, alpha helices. So remember, secondary structure describes what part of the protein forms up into an alpha helix or what part of the protein folds up into a beta sheet or a beta pleated sheet. Here, I'm showing alpha helices in this particular diagram. What kind of side chains do amino acids most likely have in this part of the protein in the middle here, though? Hydrophobic, polar, charged. Hydrophobic, remember the part that passes through the membrane in the middle here is largely going to be made up of hydrophobic amino acids because they like to interact within the hydrophobic fatty acids. Questions about that? Whoops. All right, membrane proteins are a good important part of membranes. Proteins often determine a membrane's function. And different kinds of membranes might have different proteins with different functions, but it gives the membrane then an overall different function then as well. And you can have different membranes, and we're going to talk about this when we go through cell structure. For instance, you can have the cell membrane, or what is referred to as the plasma membrane more specifically. But you can also have internal membranes inside of the cell. Like there's going to be membranes surrounding organelle structures that we're going to talk about in the next unit in the class. But organelles are membrane-bounded subcompartments inside of the cell. So organelles are membrane-bounded subcompartments of specialized function inside the cell. Can you guys think of an example of an organelle inside of a cell? Vacuole, yep. Any others? Yeah, mitochondria is going to be another. So, but a mitochondria, vacuole, they are surrounded by a membrane and then they have a specialized function. In the case of the mitochondria, its specialized function is energy processing. It's kind of the cellular powerhouse. It does parts of cellular respiration. Membrane proteins have some different functions. So, what are some different functions? Sometimes it's transport, and usually here we're talking about nutrient transport. And we will talk about examples of this. So one particular function of some membrane proteins is transport or nutrient transport. They bring stuff the cell wants into the cell. Some membrane proteins are just going to do enzymatic activity. They speed up chemical reactions. Enzymes are going to be things that speed up chemical reactions then. Some membrane proteins are going to do signal transduction. There might be some type of signal. So there might be some type of external signal or what's called a stimulus. And the cell might have a receptor. The receptor will recognize that signal. It will transmit the signal across the membrane into the cell, and then it will change cell behavior. So there are some receptor proteins that sense environmental changes, transmit the signal into the cell, and then cause the cell in a signaling cascade to change behaviors to respond to those changing conditions outside the cell. So it might be signal transduction, as some membrane proteins functioning like that. Might be cell-to-cell recognition. This is going to be, in essence, communication is another way for me to say that. You might have certain proteins on the surface of one cell communicating with proteins on the surface of another cell. And then they can actually work cooperatively together then in some cases. There's lots of proteins that are involved in intracellular joining then as well. They just attach one cell onto another, hold them together. So they anchor two cells together into things like tissues. Then those tissues, multiple cells joined together of similar function, multiple tissues can get anchored together into an organ, for instance. But you have to join the cells together and keep them together in many cases. For instance, adjacent skin cells are very tightly joined together. Very tightly joined together. Skin cells are joined so tight together that not even water molecules can pass between them. And so this is our skin, water can't pass through our skin. That keeps water out. It also keeps germs and viruses and lots of other stuff out that we don't want in our body. So these really tight intracellular joinings. And then we can also have some of these function as components of the cytoskeleton or extracellular matrix. We're going to talk more about those a little bit later on here, but they can involve an anchorage to these structures and holding the membrane to the cytoskeleton, holding the extracellular matrix to the cell, for instance. We're going to talk more about those guys a little bit later here, though. Another main part of membranes, so membranes have phospholipids. They have proteins. They also have carbohydrate components to them. So if we're looking at the components of a membrane, there's phospholipid parts, there's protein parts, there's carbohydrate parts. Membrane carbohydrates, so these are the three parts / macromolecules of membranes. Carbohydrates is kind of the third part, and they have some different functions. So for instance, carbohydrates are important for sorting cells during things like development, especially embryonic development. Carbohydrates is one way that the cells during embryonic development will join on one another, but will also sort around relative to one another. They're often involved in identification of cells and, again, in communication. So that can be an important function of these membrane carbohydrates. Membrane carbohydrates are often branched sugar chains. They're usually about 15 sugar units or 15 monosaccharides long. Also, typically carbohydrates in the membrane come in the form of either what are called glycolipids or glycoproteins. Glyco just means sugar. This basically represents sugar lipids or sugar protein is what this is translating as. So there are these hybrid molecules of protein parts and sugar parts when it comes to glycoproteins, which is pretty common. Basically a protein with a sugar attached when we're talking about a glycoprotein, or it's a lipid with a sugar chain attached when we're talking about glycolipids. Carbohydrates also, the carbohydrate components of membranes are almost always on the outside of the plasma membrane, the environmental face, in essence, of the membrane when we're talking about that. The carbohydrate groups are almost always on the outside of the cell, not on the internal face of the membrane. And different cells have different sugars that are involved. So there's a lot of variation in the sugar groups and membranes between species, even between individuals of the same species, or even between different cell types. So you guys have probably heard of blood type before. Some people have type A blood, some people have type B blood, some people have type AB blood, some people have type O blood. Probably heard of that before. Those are sugar groups on the surface of the red blood cells. So people with type A blood have type A sugars on the surface of the red blood cells. People with type O blood actually have 0 sugars on the surface of the red blood cell. Type B has B type sugars. Type AB people have both A and B sugars on their blood cells. But those are different surface markers then. Okay, so we talked a little bit so far about mosaicism. This is what I've talked about in more detail. It's a mosaic because it has phospholipid parts, protein parts, and carbohydrate parts are all part of the membrane doing different things. There's also the fluid part, and the fluid is the movement part of the fluid mosaic model. Most phospholipids in the membrane will diffuse around laterally. Laterally means what? If I was moving laterally, which way am I moving? Side to side. They'll go left, right. So when I'm saying lateral diffusion, it is side to side. But really, I'm talking about left to right is when I'm talking about this then as well. So if this is my phospholipid that I have marked, it can move around. And see here, it slid over a position and exchanged position with maybe this phospholipid. So maybe this guy slid over to the left and this guy slid over to the right, and they've changed positions. That's a lateral movement, left to right. And the components of the membrane are always doing this, constantly, very rapidly move. Lateral diffusion happens very readily, very rapidly, and it happens for phospholipids, it happens for carbohydrates, it happens for proteins, it happens for all the parts of the membrane. Transverse diffusion is what is called a flip-flop. This is where maybe my phospholipid goes from one face of the membrane, one layer of the bilayer, and it flips all the way to the other layer. That is a very rare thing. That does occasionally happen, but not very often at all. Why would a transverse diffusion be rare? Why would this phospholipid in blue very rarely move from one layer of the phospholipid bilayer to the other? First off, what are these guys down here? What are these squiggly lines? Those are fatty acids. So my inner portion from here to here, those are the fatty acids. Are those uncharged or charged? Those are uncharged. Okay. What is the little part in blue here? Do you guys think? It's a phosphocholine or a polar head group. It's a charged polar head group. What would this charged polar head group have to do to go flip from one side of the membrane and flop to the other side of the membrane? That polar head group has to pass through what region? The uncharged region. And do charged things like to interact with uncharged things? No. And so it's very difficult for this charged polar head group to flip through the hydrophobic portion of the membrane, the uncharged, and to the other side. Maybe if it starts and then it goes, I don't like it here, and it goes back. Does that make sense, everybody? So that's why transverse diffusions are very rare. But again, lateral diffusions, they don't have to do this. They just move around. They don't have to move through the hydrophobic portion of the membrane. They just move. And phospholipids will move laterally. Proteins, carbohydrates will move laterally. For instance, you could take two membranes. You can maybe take a membrane from mouse cells that have mouse proteins. You can take a portion of membrane from a human cell with human proteins. You can fuse them together, and what you end up getting is then those proteins move around over time, and instead of just staying in one spot, they end up randomly intermixed because they're constantly laterally diffusing around in these little tiny membrane bubbles. And no, I didn't literally fuse a mouse cell with a human cell. That's not what I'm showing you here. It's just a little bit, tiny bit of human membrane, little tiny bit of, not an entire cell, but mouse membrane. They've done some experiments with stuff like that. But they can see those membranes will move around and intermix evenly and randomly after about an hour or so. So proteins move laterally, phospholipids move laterally, proteins move laterally. Why is all of that important? Membrane fluidity is important. Membrane fluidity is regulated. If you do not have membrane fluidity, it is damaging to the cell. So for instance, membranes are susceptible to thermal damage. If you freeze a cell, you freeze its membranes. So for instance, in frostbite, The reason that frostbite is really bad is your cells freeze, but more specifically, the membranes freeze in place. And any kind of small pressure on them, well, then those membranes are like a thin sheet of ice. And what happens if you step on a thin sheet of ice? It cracks and breaks. And so if you press on a frozen cell, you crack the membrane and then you create cracks in it. When it unthaws, well, that membrane's disrupted, there's a big giant hole in it, and the cell just breaks apart. The membrane's not holding the stuff inside the cell anymore. There's a big giant hole in it. And that's how, for instance, frostbite kills cells. When membranes freeze in place, they're very susceptible to physical stress and physical damage, cracking them open. Too hot, well, hot is a measure of kinetic energy. So what do molecules do as they heat up? They start moving less or moving more? Moving more. Heat is a measure of kinetic energy. More heat, more movement. But the phospholipids, if they start moving around more, sooner or later they can just kind of move apart, and then you melt the membranes apart. Again, you put holes in the membrane, and then that can disrupt the membrane, and the cell breaks open, in essence. So you can, though, regulate membrane fluidity by changing the fatty acid composition on the phospholipids. Cells adjust membrane fluidity. And again, fluidity doesn't mean liquid. Fluid is a property of liquids. Liquids are malleable. They don't have a set shape. They're fluid. They're moving. Okay? Cells adjust their membrane fluidity by changing the fatty acid composition of their membranes. And as they change the fatty acids, it affects the spacing of the fatty acids. So for instance, you can use fatty acids. When I mean changing the fatty acid composition, they can use either saturated or unsaturated fatty acids. So my long, straight fatty acids are saturated ones. But my unsaturated fatty acids are any ones that put a double bonded carbon. So this is meant to represent two carbons that are joined by a double covalent bond. What's that do to change? How's that change the shape of my fatty acid though? Is it long and straight, or is it kinked and bent? Bent. What's that do to the spacing of my phospholipids? The bigger spacing. By having that bend in the hydrocarbon chain, it pushes the two phospholipids further apart. So saturated fatty acid, phospholipids with saturated fatty acids are typically crowded together. Phospholipids that use unsaturated fatty acids, they get pushed apart. They're less crowded. Does that make sense, everybody? You can use shorter or longer fatty acids. So you can use shorter chains of carbons in the fatty acids. You can use longer chains. Begin with, if you use shorter ones, you open up some molecular space. This is molecular space that's not being occupied. You can use branched versus unbranched fatty acids. So these would be unbranched here versus ones with branches coming off of it. What do the branches do to my phospholipids? Do they make them closer or further apart? Further apart. See my spacing here. Spacing is important. Structure always correlates to function. In this case, the structure correlates to the fluidity. So what do we do here? We can switch. We can use either saturated versus unsaturated, long versus short, branched versus unbranched. We can change what fatty acids are incorporated in the membrane. You guys ever notice that when it first gets cold in the winter, you are really, really cold at the very beginning of the winter? Anybody ever notice that? And then by the end of the winter, you're like, it's 35. I'll go shorts today. Why? Because you change the fatty acid composition of your membranes, and your cells are more cold tolerant, in essence. The cold doesn't bother you as much because you've acclimated to that. Okay, so what do? Heat, again, speeds up molecular motion. So as it gets hotter, things start moving more and more and more and more and more. So we're worried about the membranes melting apart. We're worried about the phospholipids in the membrane moving apart, melting apart. How do we keep the membrane from melting apart? we crowd those phospholipids in and make it hard for them to move. We put the phospholipids as close together as possible so that even when they're trying to move, they're just bumping into one another and their movement gets restricted. If I put 300 people in this room and I put you in the center, how easy would it be for you to get to the door? Not easy at all, right? You'd be constantly bumping into people, have to shove them out of the way. That's the reason there's fire codes. So what do is cell compensates with things like saturated fatty acids. We have long fatty acids. We have unbranched fatty acids. So yeah, we're going to use saturated. We're going to use long. We're going to use unbranched under high temp. The idea, though, with all of those is it crowds the phospholipids closer together. And with less molecular space, it's harder for the phospholipids to move or they're less fluid. If they're less fluid, they're less likely to melt apart. Questions about that? Cold does the opposite. With cold, it slows down molecular motion. So things are moving. As you have cold temps, that's a lack of kinetic energy. So they stop moving around until they freeze in place. How do we keep things from freezing in place? We make it easier for them to move around. So under cold temps, we want to make the phospholipids less crowded. The less crowded they are, the more they can move around. The more they can move around, the less likely they are to freeze in place. And so under cold temps, you want more molecular space so that you have more fluidity. So you're going to use unsaturated fatty acids under cold temps. You're going to use short fatty acids, and you're going to use branched fatty acids then. So that's going to be under cold temps. Again, this unsaturated pushes the phospholipids apart, and it opens up molecular space. Short fatty acids open up molecular space. Our branched fatty acids opens up molecular space. Makes the phospholipids less crowded and less likely to freeze. Questions about that then? Another component of membranes then, cholesterol. And this is present in animal cell membranes. Cholesterol is unusual, and it has some, it is unusual in the way it affects membrane fluidity. Cholesterol can restrain phospholipid movement at high temps, which is what we want. We do not want the phospholipids to be moving around at higher temps so that the membrane does not melt apart. But because of the way cholesterol works, it also can promote fluidity at low temps, interestingly enough. So cholesterol has some different properties at higher temps versus lower temperatures. Temperature affects the function of the cholesterol. And it actually, at colder temps, will promote fluidity in the membrane. And that will then actually result in the membrane being less likely to freeze in place then. So one of cholesterol's main jobs is controlling membrane fluidity at different temperatures. Questions about that? All right, membranes will be more fluid at lower membranes. At lower temperatures, how would membranes increase their fluidity? What kind of fatty acid tails are they going to use at low temps again? Unsaturated, short, and branched. So again, at lower temperatures, they're going to increase membrane fluidity by using unsaturated fatty acids, shorter fatty acids, and unbranched fatty acids. So is this membrane right here more likely to be a low temp membrane or a high temp membrane? This is probably a low temp membrane because we're seeing the phospholipids push apart. Would this be a membrane at high temp or low temp then? High temp because the phospholipids are crowded together, for instance. Does that make sense, everybody? All right, again, with that fluid mosaic model, fluidity. Its membranes are considered fluid because there's lateral diffusion of all the membrane components, and that means left, right. There is lateral diffusion of the carbohydrate components, the protein components, the phospholipids components. It's a mosaic because then it is all composed of those different parts, phospholipid parts, protein parts, carbohydrate parts. You guys know what a mosaic actually is? Have you guys been to the cafeteria where they got that mural at the end where they have all the little tiny small pictures add up to one big picture? That's a mosaic. Lots of small parts put together into a whole is in essence what we're talking about there. But the mosaic, lots of parts put together into making that membrane structure. Last thing I want to talk about with membranes is sidedness of membranes. Membranes have a sidedness to their construction. Molecules on the ends, it turns out membranes are made in the ER. You guys know what this ER stands for? Endoplasmic, I cannot write today. Endoplasmic reticulum. Think of this as a membrane factory. One of the jobs of the endoplasmic reticulum is to make membranes. Molecules that start off on the inside face of the endoplasmic reticulum end up on the outside face of the plasma membrane. And then also the endoplasmic reticulum, as well as the Golgi apparatus, we'll talk about those a little bit later. Those add either carbohydrates or proteins to the membranes. So they add these carbohydrate parts, they add these protein parts, but membranes will move from the endoplasmic reticulum, they move to the Golgi apparatus, and then eventually those little membrane bubbles fuse with the plasma membrane and can grow the plasma membrane bigger. So the membranes are made in the ER, pass through the Golgi, but they end up then getting passed over here. And as that membrane fuses with the plasma membrane gains that little bit of membrane and gets a little bit bigger. But anything that was on the inner face of the membrane ends up on the outside. Anything that was on the outside ends up on the inside, strangely enough. Then. So anything that's on the in here ends up outward facing, but fuses with the plasma membrane. Okay. Questions then about membrane structure, membrane function. All right, that's it for today. Got a little bit of a late start with computer issues. But don't forget, learning groups this week. The post-exam reflection learning group assignment, and we have Brightspace quiz on after class on Wednesday.